Bismillah ar-Rahman ar-Rahim. This is the revision for the nervous tissue data show. This picture is for the nerve cell. As you see, we have here in the center of the nerve cell the nucleus, and this is the cytoplasm, and the nerve cell, the whole boundary of the nerve cell is this one. Then we have a space, and then we have a ring of satellite cells. What are the possible questions in this picture? First of all, what is the type of this neuron? This neuron, as you see, it is smooth, it is rounded, which means that it is a unipolar. And the example for the unipolar nerve cell, you should study one example at least. It is the cells of the dorsal root ganglion or in the spinal ganglion. So it is a unipolar nerve cell, and the example is the cells present in the spinal ganglion and also in the mesencephalic nucleus of trigeminal nerve. Then you may be asked about the feature or the appearance of the nucleus of these nerve cells. The nucleus here, as you see, is large. First of all, it is here large. The nucleus is large, كبيرة, and it is spherical, rounded, spherical, and it is achromatic because it is an active nucleus. And as long it is an achromatic, it has a prominent nucleolus, which is this one. So this is the nucleolus, and this is the achromatin, or this is the remaining part of the nucleus and the nuclear envelope. So this is the feature of the nucleus of the neuron. After that, you may be asked about what are these dots present in the cytoplasm. These dots are Nessel granules. What you may be asked about the Nessel granules, what is the nature of these granules? These granules are basophilic granules that represent the following. Represent the rough ER, the free ribosomes, يعني الاتاتشد والفري، ومن ضمنهم كمان او معاهم كمان البوليزومز. يبقى هنا النسل جرانيولز هي عبارة عن basophilic granules filling the cytoplasm, and it is formed of three elements: rough ER, free ribosomes, and polysomes. What is the significance of these Nessel granules? These Nessel granules, their number varies according to the functional state and the neuronal type. Then, you may be asked also about these satellite cells. These satellite cells are belonging to what? Are belonging to the neuroglia. So the satellite cells here are belonging to the neuroglial cells. These neuroglial cells are present in the peripheral nervous system, and they are responsible for the protection of the nerve cell. So they are neuroglial cells, supportive cells, and they are present in the peripheral nervous system around the nerve cells of the ganglia. So this is a picture of a spinal ganglion. And actually they are, when you describe them, they are low cuboidal cells. Cuboidal cells. These are the satellite cells, low cuboidal cells. They are not so high, they are low cuboidal cells, not flat, they are low cuboidal cells, as you see here around this unipolar nerve cell. This is another picture showing two important structures in the nerve cell. On the right side here, you can see this is a multipolar nerve cell, and this is the nucleus. Here we can see the brown stained structures, which are the neurofibrils. The neurofibrils may be asked for their nature. These neurofibrils are actually, they are neurofilaments, which are intermediate filaments. They are actually aggregated neurofilaments, which is an intermediate filament. The intermediate filament has a diameter of 10 nanometer. But during the fixation process, the fixative cause the coagulation of these intermediate filaments called neurofilaments to stuck together and form now what is called neurofibrils. These neurofibrils, or the accumulated neurofilaments, are 2 micrometer in the diameter, so they are visible by the light microscope when we stain the slide by silver or Ag. They take a brown color. What is the function of these neurofilaments, or what we call neurofibrils now? They are called now neurofibrils because they are evident by the light microscope, so they are accumulated neurofilaments called now neurofibrils. What is the function? The function is the support. Another important thing, they are present in the body and in all processes, either dendrites or axon. In all processes, they are present, these neurofibrils, for the structural support. In the other picture on the left side, we can see these nestle granules, basophilic granules, very evident in this nerve cell. We have here the euchromatic nucleus and the nucleolus. We have the processes arising from these cells, so it is a multipolar nerve cell. Here we have a very important point to mention, which is that the nestle granules are present in the body and in the dendrites only, and they are absent in the axon. So they are not present in the axon, while the neurofibrils are present in the axon. This is a very nice picture showing the Lipofuscin pigment, which is one of the inclusions in the nerve cell. This pigment you see here is stained by golden brown color, and it takes a golden brown color. It's a normal color of it, and it represents the residue of undigested material by the lysosomes. Their amount increases with the advance of age, and they appear by this color in the nerve cell. So the more we are old, the more we have this golden brown pigment in the cytoplasm of our neurons. Here it is. This is a blood capillary, and all of these are the nerve cells, and this is the nucleus, and this is the nucleolus. So this is golden brown pigment is the Lipofuscin. You may be asked, mention another site you know that has also this Lipofuscin with the advance of age. The other side is the cardiac muscle. This is a very easy picture for a section, transverse section in the nerve trunk. We all know that the nerve trunk is formed of bundles of nerve fibers, and these nerve fibers are myelinated nerve fibers. It is supported by a connective tissue covering the whole nerve trunk called epineurium. It is a normal or an ordinary connective tissue called epineurium. And then we have another connective tissue surrounding the bundles here, there, and so on. This is called peri. Peri means around, حول, perineurium. And inside the bundles we have the myelinated nerve fibers, but they are not evident in this low power magnification. So inside these bundles we have the nerve fibers separated by the endoneurium, which is not evident in this picture. In this slide you may be asked about the nature of the epineurium and the perineurium. The epineurium is this one is a dense connective tissue covering the whole nerve, and of course its function is the support. While the perineurium, this one around the bundles, this is formed of flat epithelium-like cells, that are joined with tight junctions and this form a very important barrier. So a barrier is formed by the perineurium, very important to protect the nerve fibers. So the barrier is present in the perineurium as the cells, which are flat cells like the epithelial cells, but they are joined with tight junctions, flat epithelial-like cells joined by tight junctions, forming a barrier to protect the nerve fibers. This is also a very nice picture demonstrating the nerve trunk. We have in this picture the perineurium forming a barrier to protect the nerve fibers. We have here in between the bundles, we have in this connective tissue, we have blood vessels. Also, we have here some fat cells. The bundle itself, when we magnify it slightly, it is formed of the nerve fibers. Nerve fibers, large numbers of nerve fibers, and separated by endoneurium, but it is not evident in this picture. The nerve fibers here appear as these circles having a white content inside, representing the dissolved myelin sheath, and you can see also nuclei representing nuclei of the Schwann cells. This is also another picture for the structure of the nerve trunk. Here he is magnifying the nerve fibers. In this picture you can diagnose that we have the nerve fiber like this, and in the center we have faintly stained axon, and the area here, this area here, it should be the place of the myelin sheath, but as it dissolves during preparation of the section in alcohol, we have only here just the framework which is called neurokeratin. Neurokeratin, it is the only thing present here, but it is also not stained, and this area was occupied by the myelin sheath. And in some of the fibers you can diagnose the nucleus of the Schwann cell. So if you have a label on the dot, this dot inside, this dot inside which is faintly stained, it is the axon, faintly stained. If you have a label for the white area, it is the site of the myelin sheath which dissolved during preparation of the section. If you are asked what is now present in this area, you can answer neurokeratin. And then if you have a label for the red cycle, it is the neurolemmal sheath. Neurolemmal sheath. If you have a label for the nucleus on this red cycle, it is the nucleus of the Schwann cell. And of course you know that the neurolemmal sheath is formed by the Schwann cell, and this is the nucleus of the Schwann cell. But in order not to make a mistake in your answer, it's better to give it this circle, to give it the label neurolemmal sheath or membrane, and this is the nucleus of the Schwann cell. Here we may be asked about the Schwann cell. What do you know about, or what are the features of the Schwann cells? The Schwann cells are one of the neuroglial cells that are present only in the peripheral nervous system around the axons. What is the origin of these Schwann cells? It is ectodermal in origin. What is the function? Is the production of the myelin sheath and the electric insulation and the regeneration. So this is a higher magnification for a part of the nerve bundle, showing here these circles. These circles are the nerve fibers. Inside the circle, we have a faintly stained area, which is the axon. And this area, which is white, it is the site of the dissolved myelin sheath. And this is the outline, the red outline here, it is the neurolemmal sheath. And this is the nucleus of the Schwann cell. If you have a label on the very narrow area in between the nerve fibers, this is the endoneurium. You may be asked, what is the nature of this endoneurium? The endoneurium is formed of reticular fibers formed by the Schwann cells. يعني Schwann cells here which form the neurolimmal sheath also forms the reticular fibers of the endoneurium. The picture for the nerve trunk, but now this section allows you to see the area occupied by the myelin sheath in the nerve fibers. So it is stained by an osmic acid. We can magnify this picture. You can see here that the epineurium is stained non-specifically, and the perineurium, and these white areas or spaces here represent the endoneurium. Here you have the bundles of the nerve fibers, and they are not so evident in the picture, but I can see that they are all stained with this brown-black color. Also we have here a collection of cells which are large and filled with a black stain. Actually this is the fat cells. We have here fat cells here. These are collection of fat cells. These fat cells are filled with fat that stained black with the osmic acid. The same picture but much more magnified. You can see here that we have these bundles of nerve fibers surrounded by the perineurium. On the surface we have epineurium. They are stained non-specifically. They should take some color. And here inside the bundle you can see this whitish area representing the endoneurium. And these are fat cells full of fat stained black by this osmic acid. It is much better because here the bundles are taking the typical black, the fibers in the bundles are taking the typical black color, which is the color taken by the myelin sheath by staining with osmic acid. Also black color for the fat cells here. Here we have a bundle surrounded by the perineurium, and these fine areas here are the endoneurium, and these are collections of these nerve fibers. If you look at it much more magnified, you can see a very important thing. We can see these circles, black in color, but they are slightly thicker than the circle seen in the nerve trunk stained by hematoxin and eosine, because here they represent an area containing the myelin sheath. So here these circles are not thin, they are having a thick wall, because in this area, all this area is filled with the myelin sheath, which takes the black color of the osmic acid being formed of lipid. And the area inside here, it represents the place of the axon, which is not stained now, and of course we only have here the myelin sheath only, and we don't have the Schwann cells, nuclei, or the neurolimmal sheath. We only see the area occupied by the myelin sheath as it is fat and dissolves in the osmic acid, taking this black coloration. Also we have here the site of the endoneurium here, and we all know that endoneurium is formed of very fine reticular fibers formed by Schwann cells and not so clearly seen. Also this is the perineurium containing a barrier against any injurious material that may damage the nerve fibers. The same picture but more magnified, demonstrating the perineurium, the endoneurium, the nerve fibers stained black, demonstrating the myelin sheath, and the white area inside demonstrate the unstained axon, because the osmic acid will stain only the myelin sheath because it contains fat. Another picture also demonstrating one bundle. This is one bundle of nerve fibers present in the nerve trunk, cut transversely, and this tissue is the perineurium, acting as a barrier, and this tissue represents the site of the endoneurium, here formed of reticular fibers formed by the Schwann cells, and these are the myelin sheaths formed of these black thick-walled circles, and the white center here represents the site of the unstained axon. This slide also demonstrates the nerve bundle in a nerve trunk stained with osmic acid. And here we have very important things. We have this is the myelin present in the myelinated nerve fiber, and the white area here represents the site of unstained axon, and this is the perineurium. And these areas here around the nerve fibers, this is the endoneurium, which is formed of reticular fibers made by Schwann cells. So these are the myelinated axons, and this is the endoneurium, and this is the perineurium. In this slide, you may be asked about very important things about the myelin sheath. First of all, it is formed by, number one, formed by what? Formed by the Schwann cells. How? By making several turns around the axon. So, this is as regards the myelin sheath in the peripheral nervous system. But in the central nervous system, it is formed by other cell, which is the oligodendroglion. The Schwann cell, when form the myelin, they make a wrap, several rounded wraps around one segment in one axon. Very important, because the oligodendroglia make another action. It make wraps around one segment of many axons, but here it form several turns around one segment only of one axon. So the myelin sheath is interrupted at the nodes of Ranvier. Nodes of Ranvier. Why we have these nodes? Because the Schwann cells, during formation of the myelin sheath in the peripheral nervous system, form several wraps around one segment of a single axon. So the segments are separated from each other, and each segment is formed by one Schwann cell. You may be asked also about the functions. The functions of the myelin sheath are very easy. The myelin sheath, form an insulator around this segment so that it enhances the speed of nerve impulse through the saltatory conduction. This is a very important picture for the spinal ganglion. How can you diagnose the spinal ganglion? From the following points. First of all, the cells here are present in groups or in rows. This is very important. This is a group, and these are two rows. And this is very peculiar to the spinal ganglion, differentiating it from the sympathetic, in which the cells are scattered in the whole ganglion. After that, the cells here appear to be rounded because they are unipolar cells. They have one process that divides into two branches, one long branch to bring the sensations from the skin, and one short to carry this sensation to the spinal cord. Also here the cells are of different sizes. We have large size, we have intermediate size, and we have a small size. Each size is responsible for a certain sensation. Also here these are the most important features. If you are asked, mention one or two features that help the diagnosis. The arrangement, first of all, And number 2, the cells are variable in size. They are very evident in the picture. Also, you can add, if you are asked for 3 reasons, you can add that the cells are more or less rounded, meaning that they are unipolar cells. So, the arrangement and the size and the shape. After that, you may have a label for this one. This is the blood vessel, and all we know that the blood vessels are very minimal in the spinal ganglion. If you are asked, is there a synapse in this ganglion? No, no synapse. Why no synapse? Because the cells here are unipolar, and the cell itself brings the sensation by its own and carry this sensation to the central nervous system. So it does not depend on a synapse with another neuron. So this is no synapse in this ganglion. You may be asked, if you are diagnosed that it's spinal ganglion, you may be asked what the type of these structures. These structures are the axons of these nerve cells. These axons are myelinated. So these are myelinated nerve fibers present in between these bodies of nerve cells. This is a picture for the spinal ganglion, and here we have a covering connective tissue capsule. Actually, this capsule, it should be thicker than that of the sympathetic ganglion, and this should be taken in your consideration during drawing the ganglia. Here we have the nerve cells, and inside the cell you can see this pale structure with a dot inside. This is the nucleus, achromatic nucleus with a prominent nucleolus. And then you will notice a gap around the cell, then a circle of supportive neuroglial cells called satellite cells, described before. Here we have the nerve fibers, red and blue lines. These represent the, or pale red and dark red, these represent the myelinated nerve fibers. So we have this circle of satellite cells. This is the nerve cell, and this is the nucleus, and this, the dot here, is the nucleolus. We can magnify it more. These are the satellite cells, low cuboidal cells, neuroglial cells in the peripheral nervous system around the cells of the ganglia, and this is the nerve cell. Always there is a gap between the servant and the queen. We have this gap. And then we have this nucleus with achromatic appearance and the nucleolus, and this is the cytoplasmic granular, as you see, representing the Nessel granules. Here another picture showing many important data. Here we have the nerve cell, and this is the nucleus, and this is the nucleolus. And this cell should be in a ganglion because it is surrounded by these low cuboidal cells called satellite cells, so it is in a ganglion. Here we can see also in this cell this golden brown color of lipofuscin granules or lipofuscin pigment discussed before. Okay, here you can see that we have also in between these nerve cells, we have also these red and blue representing the nerve fibers. In this picture you can see that we have many blood vessels. Here this is one blood capillary with blood, and this is another one. Also the cells appear to be scattered in the section. The cells are not so rounded, they are angular. So most probably it is a sympathetic ganglion. This is another picture for the ganglion, but now it is stained by silver. The silver in the nerve cell will stain the Golgi apparatus and the neurofibrils by the brown color. Here you can diagnose that we have a covering capsule. We have group, row. So this is the spinal ganglion separated by myelinated nerve fibers. So this is a spinal ganglion stained by silver. This is another picture for a ganglion stained by silver, but now it is not the spinal ganglion. Why? Because we have the cells scattered in the sections, and the cells are all small in size, almost uniform in size and small. So this scattered appearance and uniform small size represent the sympathetic ganglion. What is the type of the neuron here? It is a multipolar stellate neuron. Do you have a synapse in this ganglia? Yes, there is a synapse. We have a preganglionic and postganglionic. What the type of the nerve fibers here? We have here the postganglionic fibers are unmyelinated nerve fibers. A picture for another ganglion. It is the spinal ganglion, and it is stained by silver. Why it is a spinal? Because we have a big group, then a space formed of myelinated nerve fibers, then a row, space filled with the myelinated nerve fibers, then a row, and so on. They are not scattered. We have an areas occupied by these myelinated nerve fibers, evident areas separating these groups or rows. So it is the spinal. What else? The cells here are not uniform in size. We have big size, we have small size, we have intermediate size, and each size is responsible for a certain sensation to transmit to the central nervous system. Of course, the brown coloration suggests why there is brown coloration, because the Golgi apparatus, which is well developed and fill the cytoplasm of this neuron in a position around the nucleus, is stained by silver. The Golgi apparatus here is well developed, and it is present around in the form of a network around the nucleus, is stained brown by the silver. Why we have brown coloration here in the nerve fibers? Because in the nerve fibers we have the neurofilaments, which are stained brown by silver. This is another picture showing the spinal ganglion stained by silver. How did you diagnose that it is a spinal ganglion? Because of the two things: the arrangement of the cells in the form of groups or rows. Number two, the variation in the size. We have large size, we have a small size, we have an intermediate or moderate intermediate size. Here we have also the brown coloration around the nucleus. Why? Because it stains the silver stains the Golgi apparatus, which is well developed network around the nucleus. Why we have brown coloration for these nerve fibers? The brown coloration here due to the staining of the neurofibrils 2 micrometer in diameter in the nerve fibers by the silver. And as I said before, these neurofibrils are nothing but stuck neurofilaments together to form a larger bundle due to the process of the fixation used during preparation of the slide. This is a picture for the ganglion. Why? Because we have a large collection of nerve cells. Each nerve cell is surrounded by an empty white space. These cells are stained by silver. So I should diagnose, is it a spinal or sympathetic ganglion? As the cells are almost of the same size, as the cells are angular in shape, not rounded, so they are multipolar stellate. As the cells are scattered in the section, so these are the 3 criteria by which I can make my diagnosis that it is a sympathetic ganglion stained by the silver. Why the cytoplasm is stained brown? Due to the well-developed Golgi apparatus. Why the nerve fibers here? The nerve fibers here are here appear brown due to the presence of the neurofibrils that stain brown also by silver. This is the sympathetic ganglion, also stained by silver, but of a higher magnification. You can see here that the cells have an achromatic nucleus. The cytoplasm is brown due to the Golgi apparatus well developed Here the nerve fibers due to the neurofibrils in the nerve fibers stained brown by silver. Why it is sympathetic? Because the cells are uniform in size, they are scattered in the section, they are most of them show angulation, meaning they are angle. This means that they are multipolar. Also, they have uniform size. Almost all of them are of the same size. No marked difference can be seen in between them. Start a trial exam. The wavy arrow here, this wavy arrow, points to satellite cells. The arrow itself points to the unipolar nerve cells. And this is the nucleus, and these are the granules, nestle granules present in the cytoplasm if we are asked about these granules. But this is the cell itself, which is a unipolar nerve cell. And then we have the arrowhead for the nerve fibers. These light areas and dark area represent the myelinated nerve fibers present in between the groups of nerve cells in this ganglion. This ganglion, of course, as you see, we have different sizes. So this ganglion is the spinal ganglion. And the cells also are unipolar and they are of different sizes. So this is the spinal ganglion. Identify this organ. If you see, these are multiple nerve cells with the achromatic nuclei, almost uniform size. They are almost all small. They are all scattered. So this is the sympathetic ganglion. Here, if you have a label for this cell or the arrow points to, you can say the nerve cell, the multipolar nerve cell. And you can also say it is the Golgi apparatus. So it's better to say this is Golgi apparatus, stained Golgi apparatus in a multipolar nerve cell. State the stain news, it is the silver. The wavy arrow points to nucleus. Nucleus of what? Nucleus of the Schwann cell. And the arrow itself points to this faintly stained part, which is the axon. And the arrowhead to the red circle, which is better to be mentioned as the neurolimb. Neurolimb. Not Schwann. Actually, it is a part of Schwann cell, but in order not to be in a puzzle, this is the neurolimb, and this is the axon, and this is the nucleus of the Schwann cell. This is the last picture in this trial exam. If you see here, we have number 1 here. Number 1 is representing here the body or the pericardion. Peri means around, cardion means the nucleus, so it is the body of the nerve cell. Number 1, this area, it is the body of the queen separated from the servants by this space. So this is the body of the nerve cell or the pericardion. And number 2, it is the nucleus with the prominent nucleolus. And it is, of course, an achromatic representing the high activity of this nerve cell. The number 3, these are the surrounding cells. Here they are called satellite cells. Don't be mistaken between stellate and satellite. Satellite here like a dish. Here surrounding this cell. The cell, we have different types for the shape of the cells. We have unipolar and we have a multipolar stellate. So don't make mistake between in your writing between satellite and stellate. The satellite, those are forming like a circle. Satellite, they are neuroglial cells that have been discussed before. And that's the end. Best wishes in your exam.